Hello friends, today we will learn about fermentation process. This is the second video of fermentation technology or bioprocess technology. So we'll start from overproduction of secondary metabolites. The production of secondary metabolites is more complex than primary metabolites. However, the regulatory manipulations employed for excess production of primary metabolites can also be used for the secondary metabolites as well. Several genes are involved in the production of secondary metabolites, thus, around 300 genes participate in the biosynthesis of chlorotetracycline, while 2000 genes are directly or indirectly involved in the production of neomycin. With such complex systems, the metabolic regulation is equally complex to achieve overproduction of secondary metabolites. Some regulatory mechanisms are now we will discuss. First is induction. Addition of methionine induces certain enzymes and enhances the production of cephalosporin. Tryptophan regulates ergot alkaloid biosynthesis. End product regulation. Let's know about the end product regulation. Some of the secondary metabolites inhibit their own biosynthesis, a phenomenon referred to as end product regulation. Example: penicillin, streptomycin, puromycin, chloramphenicol. It is possible to isolate mutants that are less sensitive to end product inhibition, and in this manner the secondary metabolites production can be increased. Now let's know about catabolite regulation. In this regulation process, a key enzyme involved in a catabolic pathway is inhibited or repressed by adding a commonly used substrate. Catabolic repression can be achieved by using carbon or nitrogen source. The mechanism of action of catabolite regulation is not very clearly understood. The most commonly used carbon source is glucose. It is found to inhibit the production of several antibiotics like penicillin, streptomycin, bacitracin, chloramphenicol, puromycin. The nitrogen sources, such as ammonia, also act as catabolite regulators, that is, inhibitors for the overproduction of certain antibiotics. Now, let's know about phosphate regulation. Inorganic phosphate (PI) is required for the growth and multiplication of prokaryotes and eukaryotes. Increasing PI concentration up to 1 mm is associated with an increased production of secondary metabolites. And example antibiotics like streptomycin, tetracycline, alkaloids, and gibberellins. However, very high PI concentration is inhibitory. The mechanism of action is not very clear. About the autoregulation. In some microorganisms, there occurs a self-regulation for the production of secondary metabolites. A compound designated as factor A, which is analogous to hormone, to believed to be closely involved in autoregulation for the production streptomycin by Streptomyces griseus. More such factors from other organisms have also been identified. Now, let's know about bioconversions. Microorganisms are also used for chemical transformation of unusual substrates to desired products. To desired products. This process, also referred to as biotransformation, is very important in producing several compounds. Example: conversion of ethanol to acetic acid in vinegar, sorbitol to sorbose, synthesis of steroid hormones and certain amino acids. In bioconversion, microorganisms convert a compound to a structurally related product in one or a few enzymatic reactions. The bioconversions can be carried out with resting cells, spores, or even killed cells. Non-growing cells are preferred for bioconversions since high substrate concentration can be used. Besides washing the cells easy to make them free from contamination. Sometimes mixed cultures are used for bioconversions to carry out different reactions. In recent years, the yield of bioconversions is increased by using immobilized cells at a lower cost. Now let's know about the microbial metabolic products that is high molecular weight compounds. Microorganisms are also used for the production of macromolecules that is high molecular weight compounds. Example polysaccharides, proteins including enzymes. Recombinant DNA technology has drastically improved the industrial production of various macromolecules in a cost effective manner. Now let's know about genetic improvement of strains. In general, the wild strains of microorganisms produce low quantities of commercially important metabolites. Although the yield can be increased by optimizing the fermentation conditions, the potentiality of the metabolite formation is genetically determined. Therefore, genetic improvements have to be made and new strains developed for any substantial increase in product formation in a cost effective manner. There are strain development programs to increase the product yield by 100 times or even more. The nature of the desired product determines the success associated with strain improvement. For example, if alterations in one or two genes can improve the product yield, it is simpler to achieve the target. This type of approach is sometimes possible with primary metabolites. As regards the secondary metabolites, the product formation and its regulation are quite complex. Hence, several genetic modifications have to be done to finally produce high yielding strains. Ideally speaking, the improved strains should possess some characteristics to finally result in a high product formation. Let's know about the characteristics. First characteristic is shorter time of fermentation. Second is capable of metabolizing low cost substrates. Third is reduced oxygen demand. Fourth is decreased foam formation. Fifth is non-production of desired compounds. Sixth is tolerance to high concentration of carbon or nitrogen sources. Seventh is resistant to infections of bacteriophage. It is always preferable to have improved strains of microorganisms which can produce one metabolite as the main product. In this way, the production can be maximized and its recovery becomes simpler. Through genetic manipulations, it has been possible to develop strains for the production of modified or new metabolites which are of commercial value. Example, modified or newer antibiotics. The major limitation of strain improved is that for most of the industrially important microorganisms, there is a lack of detailed information on the genetics and molecular biology. This hinders the new strain development. Now let's know about the methods of strain development. There are three distinct approaches for improvement of strain mutation, recombination and recombinant DNA technology. Now let's know about the mutation. Any change that occurs in the DNA of a gene is referred to as mutation. These mutations result in a structural change in the genome. Mutations may be spontaneous or induced by mutagenic agents. The spontaneous mutations occur at a very low frequency and usually are not suitable for industrial purposes. Mutations may be induced by mutagenic agents such as ultraviolet light, various chemicals like nitrous oxide, guanidine, hydroxylamine, site-directed mutagenesis is also important for strain improvement. Now let's know about selection of mutants. Selection and isolation of appropriate mutant strains developed is very important for their industrial use. Two techniques commonly employed for this purpose. First is random screening, second is genetic recombination. Let's know about the random screening. The mutated strains are randomly selected and checked for their ability to produce the desired industrial product. This can be done with model fermentation units. The strains with maximum yield can be selected. Random screening is costly and tedious procedure, but many at a times this is the only way to find the right strain of mutants developed. Now let's know about the selective isolation of mutants. There are many methods for selective isolation of improved strains. First is isolation of antibiotic resistant strains. The mutated strains are grown on a selective medium containing an antibiotic. The wild strains are killed while the mutant strains with antibiotic resistance can grow. Such strains may be useful in industries. Second is isolation of anti-metabolite resistant strains. Antimetabolites which have structural similarities with metabolites can block the normal metabolic pathways and kill the cells. The mutant strains resistant to antimetabolites can be selected for industrial purposes. Third is isolation of autotrophic mutants. An autotrophic mutant is characterized by a defect in one of the biosynthetic pathways. As a result, it requires a specific compound for its normal growth. For instance, dire mutants of Corynebacterium glutamicus require tyrosine for their growth while they are accumulate phenylalanine. The isolation of such mutants can be done by growing them on a complete agar medium that can specifically support the biochemically defected mutant. Now let's know about genetic recombination. The strain improvement can be made by combining genetic information from two genotypes by a process called genetic recombination. The recombination can be brought out by transformation, transduction, conjugation and protoplast fusion. There are many advantages of genetic recombination. By crossing high product yielding mutant strains with wild type strains, the fermentation process can be further increased. Second is different mutant strains with high yielding properties can be combined with recombination. Third is this is gradual decline in the product yield after each stage of mutation due to undesirable mutations. This can be prevented by using recombination. Now let's know about the principles of microbial growth and culture systems. The growth of microorganisms is a highly complex and coordinated process, ultimately expressed by increase in cell number or cell mass. The process of growth depends on the availability of requisite nutrients and their transport into the cells. The environmental factors such as aeration, oxygen supply, temperature, and pH. Doubling time refers to the time period required for doubling the weight of the biomass, while generation time represents the period for doubling the cell numbers. Doubling times normally increase with increasing cell size and complexity. Bacteria, it should be 0.302 in 1 hour, yeast 1 to 2 hour, animal cells 25 to 48 hours, plant cells is 20 to 70 hours. In general, when all other conditions are kept idle, growth of the microorganisms is dependent on the substrate supply. The microorganisms can grown on batch, feed batch, semi-continuous or continuous culture systems in a bioreactor. In batch fermentation, the growth medium containing the substrate is inoculated with microorganisms and the fermentation proceeds without the addition of fresh growth medium. In phase batch fermentation, substrates are added at short time intervals during fermentation. In batch and feed fermentation, the growth of the cells is quite comparable, and in both cases, the growth medium is not removed until the end of the fermentation process. In case of continuous fermentation, as fermentation proceeds, fresh growth medium is added continuously. Simultaneously, an equal volume of spent medium containing suspended microorganisms is removed. This enables the cells to grow optimally and continuously. Now let's know about the batch culture or batch fermentation. A batch fermentation is regarded as a closed system. The sterile nutrient culture medium in the bioreactor is inoculated with microorganisms. The incubation is carried out under optimal physiological conditions, pH, temperature, oxygen supply, agitation, etc. It may be necessary to add acid or alkali to maintain pH and anti-foam agents to minimize foam. Under optimal conditions for growth, some six typical phases of growth are observed in batch fermentation. First is lag phase, acceleration phase. Third is logarithmic phase, or we can say it as exponential phase. Fourth is a deceleration phase, and fifth is stationary phase, and sixth is death phase. Now let's know about lag phase. The initial brief period of culturing after inoculation is referred to as lag phase. During the lag phase, the microorganisms adapt to the new environment, available nutrients, pH, etc. There is no increase in the cell number, although the cellular weight may slightly increase. The length of the lag phase is variable and is mostly determined by the new set of physiological conditions and the phase at which the microorganisms were existing when inoculated. For instance, lag phase may not occur if the culture inoculated is at exponential phase and growth may start immediately. Now let's know about acceleration phase. This is a brief transient period during which let's know about acceleration phase. This is brief transient period during which cells start growing slowly. In fact, acceleration phase connects the lag phase and log phase. Now let's know about log phase. Now let's know about log phase. The most active growth of microorganisms. Log phase. The most active growth of the microorganisms and multiplication occur during log phase. The cells undergo several doublings and the cell mass increases. When the number of cells or biomass is plotted against time on a semi-logarithmic graph, a straight line is obtained, hence the term log phase. Growth rate of micro Growth rate of microbes in log phase is independent of substrate concentration as long as excess substrate is present and there are no growth inhibitors in the medium. In general, specific growth rate of microorganisms simpler, simpler substrates is greater than for long chains. This is explained on the basis of extra energy needed to split long chain substrates. Two log phases are observed when a complex nutrient medium with two substrates is used in fermentation, and this phenomenon is referred to as diaxy. And this happens since one of the substrates is preferentially metabolized first, which represses the breakdown of second substrates. After the first substrate is completely degraded, second lag phase occurs, during which period the enzymes for the breakdown of second substrate the synthesis is synthesized. Now a second log phase occurs. Now let's know about deceleration phase. As the growth rate of microorganisms during log phase decreases, they enter the deceleration phase. This phase is usually very short-lived and may not be observable. Now let's know about second stationary phase. As the substrate in the growth medium gets depleted and the metabolic end products that are formed inhibit the growth, the cells enter the stationary phase. The microbial growth may enter slow down or completely stop. The biomass may remain almost constant during stationary phase. This phase, however, is frequently associated with dramatic changes in the metabolism of the cells, which may produce compounds of biotechnological importance, that means production of antibiotics. Now, let's know about the death phase, the last phase. This phase is associated with cessation of metabolic activity and depletion of energy reserves. The cells die at an exponential rate. In the commercial and industrial fermentation, the growth of the microorganism is halted at the end of the log phase or just before the death phase begins and the cells are harvested. Now let's know about the feed batch culture or feed batch fermentation. Feed batch fermentation is an improvement of batch fermentation wherein the substrate is added in increments at different times throughout the courses of fermentation. Periodical substrate addition prolongs log and stationary phases which results in an increased biomass. Consequently, production of metabolites during stationary phase is very much increased. As it is difficult to directly measure substrate concentration in feed batch fermentation, other indicators that correlate with substrate consumption are used. The formation of organic acids, production of carbon dioxide, and changes in pH may be measured and accordingly substrate addition carried out. In general, feed batch fermentation requires more careful monitoring than batch fermentation and is therefore not a preferred method by industrial biotechnologists. Now let's know about the feed batch fermentation for the production of recombinant proteins. In recent years, feed batch fermentation has become popular due to very high yield for the production of recombinant proteins. Depending on the microorganism and the nature of recombinant protein, the feed batch fermentation can increase the product yield from 25% to 1000% compared to batch fermentation. Careful monitoring of the fermentation reaction and appropriate addition of substrate, that means carbon and nitrogen sources and trace metals, substantially increases the product yield. Now let's know about the limitations. The major limitation of feed batch fermentation is that the microorganisms in the stationary phase produce proteolytic enzymes or proteolases. These enzymes attack the recombinant proteins that are being produced. By carefully monitoring the fermentation, the lag phase can be prolonged. The onset of stationary phase is delayed. By this way, the formation of proteases can be minimized. Now, let's know about the feed batch cultures. For higher organisms, feed batch cultures are successfully employed for mammalian and insect cells. This is very advantageous for the production of human therapeutic proteins with good yield. Now let's know about semi-continuous culture or semi-continuous fermentation. Some of the products of fermentation are growth linked. And such products are formed at the end of the log phase. Example, ethanol production. In semi-continuous fermentation, a portion of the culture medium is removed from bioreactor and replaced by fresh medium. This process of culture medium change can be repeated at appropriate intervals. In the semi-continuous fermentation, the lag phase and the other non-productive phases are very much shortened. The product output is much higher compared to batch culture systems. Semi-continuous fermentation technique has been successfully used in the industrial production of alcohol. There are, however, certain disadvantages of semi-continuous fermentations. These include the technical difficulties of handling bioreactors, long culture periods that may lead to contamination, mutation, and mechanical breakdown. Now let's know about the continuous culture or continuous fermentation. Continuous fermentation is an open system. It involves the removal of culture medium continuously and replacement of this with a fresh sterile medium in a bioreactor. Both addition and removal are done at the same rate so that the working volume remains constant. Further, to maintain a steady state condition in continuous process, it is advisable that the cell loss as a result of outflow is balanced by growth of the organisms. Now let's know about the homogeneously mixed bioreactors. In this type, the culture solution is homogeneously mixed and the bioreactors are of two types. Chemostat bioreactors. The concentration of any one of the substrate, carbohydrate, nitrogen source, salts, oxygen is adjusted to control the cell growth and maintain a steady state. Turbidostat bioreactors. Let's know about turbidostat bioreactors. In this case, turbidity measurement is used to monitor the biomass concentration. The rate of addition of nutrient solution can be appropriately adjusted to maintain a constant cell growth. Now let's know about plug flow bioreactors. In plug flow bioreactors, the culture solution flows through a tubular reaction vessel without black mixing. The composition of the medium, the quantity of cells, oxygen supply and product formation vary at different locations in the bioreactor. Microorganisms along with nutrient medium are continuously added at the entrance of the bioreactors. Now let's know about the industrial application of continuous fermentation. Continuous fermentation process have been used for the production of antibiotics, organic solvents, single cell protein, beer and ethanol besides wastewater treatment. Now let's know about advantages of continuous fermentation. The size of the bioreactor and other equipment used in continuous fermentation are relatively smaller compared to batch fermentation for the production of same quantity of product. Now let's know of the product. Second point is the yield of the product is more consistent since the physiological state of the cells is uniform. The downtime between two successive fermentation for cleaning and preparing the bioreactor for reuse is avoided in continuous fermentation. Continuous fermentation can be run in a co-effective manner. Disadvantage of continuous fermentation. Despite many advantages of continuous fermentation, it is not very widely used in industries. Some of the drawbacks are now we will discuss. First is continuous fermentation may run continuously for a period of 500 to 1000 hours. Maintenance of sterile conditions for such a long period is difficult. The recombinant cells with plasmid constructs cannot function continuously and therefore the product yield decreases. It is not easy to maintain the same quality of the culture medium for all the additions nutrient variations with alter the growth and physiology of the cells and consequently the product yield. In addition to the disadvantage till now we discussed, industrial biotechnologists are rather reluctant to switch over to continuous fermentation from the batch fermentation. However, it is expected that continuous fermentation will also become popular in due course. Now let's know about growth kinetics of microorganisms. In the different types of fermentation processes, batch, feed batch, semi-continuous and continuous. Now we discussed, already discussed, the kinetics of microbial growth with special reference to log phase of a batch fermentation. We will discuss now. After completion of lag phase, the cell enters log phase, which is characterized by exponential growth. If the initial number of the cell is n0, then after first generation the cell number will be n0 into 2 to the power 1. After the second generation the cell number will be n0 into 2 to the power 2. After third generation n0 into 2 to the power 3 and so on. Thus the number of cells after a given time will be nt equals to n0 into 2 to the power n, where n is the number of generations. The term doubling time or mean generation time refers to the time taken for doubling the cell number or biomass. The specific growth rate constant expresses by omega is the direct manure measure of the rate of growth of the organism. If n is the number of cells at a given time, then the increase in the number of cells with time is given by the formula dn by dt equal to mu n. If x is a biomass concentration at a given time, then the increase in the biomass with time is given by dx by dt equals to mu x. In general, the specific growth rate, that is mu, is a function of the concentration of limiting substrate S. The maximum specific growth rate is mu max and a substrate specific constant Ks. The relationship was expressed by Monod by reaction mu equals to mu max S divided by Ks plus S. Both S and Ks are expressed as concentration, example in moles or grams per liter. The growth rate of an organism is not fixed, but it is variable depending on the environmental conditions such as concentration of substrate and temperature. At a low concentration, the substrate is limiting factor for growth. The batch culture, the substrate is initially present at a higher concentration. Example, S is greater than Ks. Hence, the equation 3 is approximately 1. S divided by Ks plus 1 equals to 1. Thus, mu equals to mu max. Now let's know about the classification of fermentation processes. There are different ways of classifying the fermentation process. The major classification as batch, feed, batch, semi-continuous and continuous fermentation processes are already we discussed. Another classification based on the product formation in relation to energy metabolism we will discuss now. Type 1 fermentation. When the product is formed directly from a primary metabolism used for energy production, it is referred to as a type 1 and may be represented as substrate A gives rise to product. Substrate A gives rise to B, gives rise to C, gives rise to gives rise to C, gives rise to D, gives rise to product. Growth energy metabolism and product formation almost run in a parallel manner. In this type, trophophase and iodophase are not separated from each other. Example, production of ethanol, gluconic acid and single cell protein. Type 2 fermentation. Let's know about type 2 fermentation. Type 2 category, the product is also formed from the substrate and used for primary energy metabolism. However, the product is produced in the secondary pathway. Substrate A gives rise to B, gives rise to C, gives rise to D, primary metabolism. A gives rise to E, E gives rise to F, gives rise to G, gives rise to product. At the beginning, the growth of the microorganism is accompanied by high substrate utilization with little or no product formation. Now the growth is slowed down, but the substrate consumption is high, and this is coupled with product formation. Type 2 fermentation. In type 2 fermentation, the trophophase and the iodophase are separate. Production of some amino acids, citric acid and itaconic acid are good examples of type 2 fermentation. Now let's know about type 3 fermentation. This is a clear distinction between the primary metabolism and product formation in type 3 fermentation, as they occur at separate times. Substrate consumption and rapid growth occur in the first phase, and the product formation occurs in the second phase. This product is formed from amphibolic metabolic pathways and not from the primary metabolism, example production of vitamins and antibiotics. Now let's know about the overlap of different types of fermentation. Type 1, type 2 and 3 fermentations originally categorized by Garden in 1959. There are intermediate forms based on the composition of nutrient culture medium, strain of the microorganism used and product formation. For instance, industrial production of lactic acid falls between type 1 and type 2, while production of the antibiotic amyloglycoside is intermediate between type 2 and type 3. It is sometimes difficult to categorize the industrial fermentation under any of these types due to complex nature of the process. Example, mycelium producing microorganisms in relation to antibiotic production. Now let's know about the fermentation process. The fermentation process basically consists of inoculum preservation, inoculum buildup, pre-fermenter culture and finally production fermentation. Now first we will discuss about inoculum preservation or culture maintenance. The preservation of high yielding strains of microorganisms for fermentation is very important for product formation in substantial amounts. The ultimate purpose of preservation is to maintain the strain as long as possible. Without cell division, there are different methods of preservation. First is storage at low temperature, that means 2 to 6 degrees centigrade. In this method, the microorganisms can be stored in a refrigerator, in liquid culture or a stab culture. Although this is the easiest method of preservation, this is a high risk of contamination. Storage by freezing. Let's know about storage by freezing. The microbial cultures can be frozen and preserved for several years. This in the freezer. The preservation can be done at minus 18 degrees centigrade or at minus 80 degrees centigrade. For the preservation at minus 196 degrees centigrade, liquid nitrogen must be used. It is very important that the freezing is done slowly to prevent damage and killing of the microorganism. If proper care is not taken, as many as 95% of cells may be killed by freezing and thawing. Storage by lyophilization. Let's know about storage by lyophilization. Preservation of microorganisms by lyophilization, that is freeze drying, is the best method, although it requires special equipments. In fact, lyophilization is the method of choice by many fermentation biotechnologists. The storage of microorganisms can be done by any one of the three techniques till now is discussed. The storage of microorganisms can be done by any one of three techniques till now we discussed. However, for each method, optimal conditions for preservation must be worked out for each strain separately. In general, the preserved master strains are cultivated once in 2 years for checking of their activity. When needed for use, the working strain can be obtained from the master strains. Now let's know about inoculum buildup. The preserved cultures have to be revived for their industrial use. This can be done by growing the cultures in liquid or on solid media. The actual process and the conditions used for inoculum build-up largely depend on the when needed for use, the working strains can be obtained from the master strains. Now let's know about inoculum buildup. The preserved cultures have to be revived for their industrial use. This can be done by growing the cultures in liquid or the semi-solid or on solid media. The actual process and the conditions used for inoculum buildup largely depend on the preservation technique used. There are wide variations in the growth times which depend on the type of preservation and the organisms used, that is refrigerated cultures in 2 to 6 degrees centigrade, bacteria 6 to 24 hours, actinomycetes, that is 1 to 3 days, fungi 1 to 5 days, and frozen cultures, that is 18 degree centigrade to 18 degrees centigrade or minus 80 degrees centigrade or minus 196 degrees centigrade. Bacteria, that is 6 to 48 hours. Actinomycetes, that is 1 to 5 days, and fungi should be in 1 to 7 days. Now let's know about lyophilized cultures. For all organisms, 4 to 10 days for proper growth and to obtain sufficient quantity of inoculum, a series of cultures are prepared. For good fermentation yield, the number of cells and spores, nutrient medium, temperature and age of the inoculum are important. In the inoculum buildup is suspended in a surface active agent such as twin 80 and transferred to the bioreactor for fermentation. Now let's know about fermenter culture. Fermenter culture, pre-culture or fermenter culture is often required for inoculating large size bioreactors. Inadequate quantity of inoculum will not only delay the product formation but also reduce the yield drastically. By culturing the microorganisms in small fermenter, the synthesis of the inoculum can be increased for large scale industrial use. Biotechnologists have worked out the requisite inoculum concentration for optimal fermentation. Example, for bacterial fermentation, the inoculum concentration should be between 0.2 to 3.0%. For fungal fermentation, it is in the range of 5 to 10%. Now let's know about production fermentation. The general features and the different types of bioreactors are already till now we discussed. The size of the fermenter used mainly depends on the product. For example, a small bioreactor can be used for producing diagnostics, enzymes and substances for molecular biology by recombinant microorganisms, while large bioreactors are employed for producing single cell protein and amino acids. For appropriate production of by fermentation, several parameters need to be carefully considered and optimized. These include composition of nutrient medium, carbon and nitrogen sources, batch to batch variations, effect of relation on nutrients under the on pH, alteration in temperature and aeration. The parameters temperature, pressure, aeration and stirring now we'll discuss. First is temperature. The temperature must be so maintained that there occurs maximal growth of microorganism with optimal product formation. Although this is not always possible, in general there are two temperature ranges to run the fermentation: a mesophil range, that is 20 to 45 degrees centigrade, and a thermophil range which should be below 45 degrees centigrade. Sometimes two different temperatures are used for the same fermentation process. A higher temperature is employed for good growth and then the temperature is decreased for optimizing product formation. Now let's know about the pressure. Appropriate maintenance of hydrostatic pressure, particularly in large sized bioreactors, is very important. This is because pressure influences the solubility of oxygen and carbon dioxide in the culture medium. An overpressure in the range of 0.2 to 1.5 bar is generally used. Aeration. Let's know about the aeration. A bioreactor gets aerated by the supply of oxygen and therefore adjustment must be made to furnish required amount of oxygen to the microorganisms. Usually the aeration rate is in the range of 0.25 to 1.25 bbm volume of air. Now let's know about steering. The type and the speed of impellers determines the steering rate in a fermenter. In general, the impeller speed decreases as the size of the fermenter increases. Thus, for a small bioreactor size 1 to 20 liter, the impeller speed is in the range of 250 to 350 rpm. While for the large bioreactor size around 450 liter, the impeller speed is 60 to 120 rpm. Root per. Now let's know about measurement and control of bioprocess parameters. Now let's know about measurement of and control of bioprocess parameters. There are a large number of physical, chemical and biological parameters that can be measured during fermentation or bioprocessing. For data analysis and appropriate control, some special sensors have been developed to carry out measurement in the bioreactors. The basic requirement of all the sensors is that they must be sterilizable. The measurements of the parameters can be done either directly or in the bioreactor or in the laboratory. Now let's know about pH measurement. There are pH electrodes that can withstand high temperature, that is sterilization, pressure and mechanical stresses, and yet measure the pH accurately. Combination electrodes are being used. In fact, electrodes are also available for measuring several other inorganic ions. Oxygen and carbon dioxide measurement. Oxygen electrodes and carbon dioxide electrodes can be used to measure oxygen and carbon dioxide concentrations respectively. The electrodes are amperometric in nature. They are, however, susceptible for damage on sterilization. In a commonly used technique, oxygen and carbon dioxide respectively can be measured by magnetic property of oxygen and the infrared absorption of carbon dioxide. This can be done by using sensors. Use of mass spectrometer. Let's know about the use of mass spectrometer. The mass spectrometer is a versatile technique. It can be used to measure the concentration of nitrogen, NH3, ammonia, ethanol and methanol simultaneously. In addition, mass spectrometer is also useful to obtain information on quantitative and qualitative exchange of oxygen and carbon dioxide. Now let's know about the use of gas permeable membranes. The measurement of dissolved gases up to 8 simultaneously can be done almost accurately by using gas permeable membranes. The advantage is that such measurement is possible to carry out in the nutrient medium. Let's know about the use of computers. Computers are used in industrial biotechnology for data acquisition, data analysis and developing fermentation models. Let's know about data acquisition. By employing line sensors, the computers and by employing online sensors and computers in fermentation system, data can be obtained with regard to the concentration of oxygen and carbon dioxide, pH, temperature, pressure, viscosity, turbidity, aeration rate, etc. Certain other parameters can be measured in the laboratory, that is offline measurements. The information collected from online and offline measurements can be entered into a computer. In this fashion, the entire data regarding a fermentation can be processed, stored and retrieved. Now let's know about the data analysis. The data collected on a computer can be used for various calculations. Example, rate of substrate utilization, rate of product information, rates of oxygen uptake and carbon dioxide formation, heat balance, respiratory quotient. Through computer data analysis, it is possible to arrive at the optimal productivity for a given fermentation system. Now let's know about development of fermentation models. The computer can be used to develop mathematical models of fermentation process. These models in turn will be useful to have a better control over fermentation systems with high productivity in a cost effective manner. In general, now let's know about the scale up. In general, fermentation of bioprocess techniques are developed in stages starting from laboratory and finally leading to an industry. The phenomenon of developing industrial fermentation process in stages is referred to as scale up. Scale up is necessary for implementing new fermentation technique developed and using mutant organisms. The very purpose of scale up is to develop optimal environmental and operating conditions at different levels for a successful fermentation industry. The conditions that need to be studied include substrate concentration, agitation and mixing, aeration, power consumption and rate of oxygen transfer. In a conventional scale-up, a fermentation technique is developed in 3 to 4 stages. The initial stage involves a screening process using petri dishes or Erlenmeyer flask. The next stage is a pilot project to determine the optimal operating conditions for a fermentation process with a capacity of 5 to 200 liters. The final stage involves the transfer of technology developed in the laboratory to industry. It has to be continuously noted that a fermentation process that works well at the laboratory scale may work poorly or may not work at all on industrial scale. Therefore, it is not always possible to blindly apply the laboratory conditions of a fermentation technique developed to industry. At the laboratory scale, one is interested in the maximum yield of the product for unit time. At the industry level, besides the product yield, minimal operating cost is another important factor for consideration. An industrial biotechnologist has to apply intellect and in technical skill for establishing a successful fermentation setup in a cost effective manner. Now let's know about the food technology. Food technology is a vast field, the silent feature of food processing which has some relevance to bioprocess engineering and technology are now we will discuss. In general, the food processing is carried out by achieving some objectives. First is for the purpose of storage and transport. Second is to protect from contamination. Third is to increase the shelf life. Fourth is to make it attractive for consumers. For appropriate processing of food, many criteria need to be taken into account. These include the ability of the microorganisms and the paste to invade and grow on foods and the chemical instability and biological activity of foods. Now let's know about the food preservation. Among the many food processing techniques, food preservation is the most important one. Selected food preservation process are now we will discuss. First is drying. Exposure of foods to sunlight and drying is a natural and an earliest method of food preservation. Drying involves the removal of water, consequently, the moisture content of the food falls and the invading microorganisms cannot grow. Hot air is also frequently used to remove moisture. In actual price, foods are dried by using different types of equipments. Swallow bed dryer. Drying is carried out in perforated conveyor belts. Deep bed dryers. Drying occurs in bins. That is deep bed dryers. Spray dryers. That is to dry liquid foods and food slurries. Freezing dryers. That is to dry soup ingredients, beverage extracts. Now let's know about chilling. The techniques of chilling is commonly used to preserve food without freezing. The process of chilling is carried out around 0 degrees centigrade with humidity between 85 to 95%. The shelf life of fresh foods like meat, fish and dairy products can be extended by chilling. It is reported that if chilling is done under low oxygen conditions, the shelf life of foods increases. Chilling is frequently used for long distance shipping of meat, apples, vegetables, etc. Now let's know about freezing. Foods can be preserved in a frozen state, that is minus 30 degrees centigrade to minus 10 degrees centigrade. At a very low temperatures, the water activity and the reactant mobility are low. Hence, the chemical reactions are reduced. Foods can be frozen by using a stream of refrigerated air or by passing inert refrigerant or cryogenic gases. Rapid freezing is advocated to minimize the adverse effects on the texture of foods. Now let's know about thermal processing. By applying heat comparable to sterilization, majority of the microorganisms and the spores present in the foods can be killed. Thermal processing is carried out in sealed containers devoid of oxygen and thus aerobic organisms cannot grow. Further, higher temperature also leads to inactivation of enzymes and consequently microbial growth will be retarded. Now, bioprocess technology or fermentation technology finally finished. This is a vast course, but there are many things to learn. In two videos, it is not possible to give all the contents. And till now, any questions regarding this, you can ask me in the comment box. And please subscribe my channel to know more about biotechnological new latest technologies. Thank you.